Okay, it's 10:30 Iowa time, so time to get started. Welcome, everybody. My name is Jack Deckers. I'm a faculty member in animal breeding and genetics at Iowa State University and a co-PI on the Agricultural Genome to Phenome Initiative. And this is our 6th field day. I'm glad to see a number of people online, and I realize that number may increase as we go, and it's recorded, so others will probably view the recording because of other time commitments. So today we're going to learn about aquaculture, and I'm very excited about this because I don't know too much about aquaculture, and I think the same holds for most of the people involved with the AG2PI initiative. But aquaculture is a big part of agriculture production, a growing part of agricultural production. And so we're going to hear about that today. If you have questions, we have 3 speakers. If you have questions, please put them in the chat and send it to everyone. And then if appropriate, I'll ask the question during the talk, but otherwise we'll have a pause after every presentation, and we'll follow up on the questions. So our first speaker is Dr. Caird Rexroad. He's a program leader at USDA ARS in animal production and protection. He's a national program leader in animal production and protection, and with a focus on aquaculture. And he's going to introduce the field of aquaculture. And then he will also introduce our 2 speakers who will focus on shellfish and finfish. So Caird, I'll turn it over to you if you want to start sharing your screen.
Fantastic, Jack. Thank you very much. I'll go ahead and share my screen. Okay. So I, first of all, I want to say thank you, Jack, to you and the organizers for dedicating one of the field days to the subject of aquaculture. So it really is a pleasure to be able to talk a little bit about aquaculture. As you said, I will introduce the topic, and then we have some great speakers who are going to follow up talking a little bit about both shellfish and finfish aquaculture in significantly more detail. So aquaculture can mean many things to different people. In general, we're talking about propagating, rearing of aquatic species in controlled, or what we say, select environments, or simply farming the waters for diverse products and services. Many times the controlled environment can be indoors, such as land-based recirculating aquaculture systems. It can be outdoors. It can be net pens in open waters. And so sometimes we say select environments. Obviously, in open waters, we're not controlling the environments, but we are dedicating that space for aquaculture production. And so that's how we use that term. Coexisting with fisheries, so aquaculture and fisheries are different things. Fisheries, we're typically talking about, the natural populations and managing wild caught harvests, where aquaculture, we're talking about farm production. We are talking about many different kinds of species. I think worldwide there are over 600 some species that are cultured in the U.S. The numbers range anywhere from 200 to 300 species that we have in production. And we're talking about aquatic animals, aquatic plants, and including the algal species, the seaweeds, the macroalgae that can be reared in marine environments. And as been stated, this is the fastest growing global agricultural industry. So aquaculture has many uses. USDA, we're primarily focused on food production. There are also many state and federal agencies and others interested in rebuilding stocks of species that are threatened or endangered, providing ecosystem services, mostly in the case of shellfish or seaweeds, where we have systems for removing nutrients from the water, enhancing recreational and commercial activities. Most states and the federal hatchery systems support recreational angling, providing live organisms for aquaria enthusiasts. So all of the fish that you see in the pet stores that you can bring home and keep in an aquarium. And then finally, being a potential new source of biofuels. And here we're talking mostly about seaweeds and the ability to produce biofuels. So this is a slide that you'll see in many aquaculture talks. We all use it. It comes from the FAO. And what we're showing here is the ability to get seafood from capture fisheries versus aquaculture. And in this case, separating out both the inland capture from the marine, and then also the aquaculture, what's inland production and versus the marine environment. And so what has happened as a trend over the last many decades is that we're seeing about half of our seafood that's available for food is coming from aquaculture production. And this is true as a global statistic. Another trend is that we're monitoring our fisheries and making sure that they're sustainable. And we see also a trend where we're seeing more and more of our fisheries that are unsustainable, and the fisheries that we have that were underfished are also in decline. So all of this, global statistics in support of more aquaculture production to meet the demand for seafood. There are many production systems for aquaculture, and I'm showing some of the examples here in this slide. In the upper right is what we call integrated multi-trophic aquaculture. This is kind of a combination system where you have a net pen or some kind of system holding fish that you're feeding. There might be seaweed or shellfish also in that system that are using some of the nutrients from the fish aquaculture to support the other forms of aquaculture. And so when you have all of this in combination, you can supply all three of these types of organisms. The second here that I'm showing is a recirculating aquaculture system. On both sides of this infrastructure, you have tanks, those are where the fish are held. But then in the middle, you have a series of filters and systems for disinfecting water, removing both liquid and solid waste so that you can use the water again for the fish production. And so what's great about this is it allows you to have fish production just about anywhere where you can supply the initial source of water. Recirculating systems tend to recirculate about 99%. And then finally on the bottom, I'm showing two different kinds of pond production. We have traditional ponds are on the outside. These are just large ponds where we're raising the fish. You might use aerators to supplement the oxygen in the water. The two internal ponds are what we call the split pond system or the partitioned aquaculture system. And in this case, the fish are actually raised in about 15% of the volume of water in the pond, and the rest of the pond uses the natural flora and fauna to treat the water, remove the waste, and then you can circulate the water back in through the fish part of the system. And so there's a lot of diversity in the species that are raised. There's also a lot of diversity in the kinds of systems that are used for production. So a little bit on the status of U.S. seafood. We actually rank 17th in the U.S. for global aquaculture production, far behind countries like China, who's leading the world at 62%. About 90% of what Americans eat is imported. So it's a huge trade deficit. And so a lot of opportunity to use our natural resources to meet domestic demand through domestic supply. Only about 5% of the seafood that we eat is coming from our own aquaculture production. We have about $1.5 billion industry. So that's all of aquaculture, including all the catfish, all the trout, and so on. That is not including recreational fisheries and those activities. So I think it's always really important to note U.S. consumers are using discretionary funds to purchase seafood. And so they have many choices and they're looking at price and quality, safety issues, and issues related to the sustainability of production. In this slide, I just want to show again the diversity. This is the number of species, and this is from the Census on Aquaculture that always follows the Census of Agriculture conducted by the National Ag Statistics Service. You can see that blue and channel catfish continue to be the leading finfish production, oysters on both coasts, and I won't go through the whole list, but you get the idea that there's many species that are being produced. If you look on the bottom right, there are many species where there's only one or few companies that are participating, and so they can't be included in the survey data that are presented, but still important industries, and in many cases an opportunity for new production. So to look at some of the opportunities for the industry, so the dietary guidelines call for Americans to double their seafood intake. We have Asia becoming a net importer of seafood, so other countries are looking to them for to export their products, which means that things that are currently being imported by the U.S. Could be met through domestic production. U.S. is a major producer of fish feed ingredients and primarily soybeans, but being a producer of fish feed ingredients gives us an opportunity to keep some of those at home for our own production. In terms of where we would have aquaculture, we have the Great Lakes, farmed to some degree by Canada, not farmed by the U.S. We have land-based opportunities, such as those recirculating aquaculture systems, which would facilitate production in any kind of rural or urban or other environment. And then we have this U.S. exclusive economic zone that we could use for aquaculture. Not currently using it in production. These are federal waters. And last year, there was an executive order that assigned NOAA the task of figuring out where we could have sustainable aquaculture production in federal waters. So some of the industry challenges, I think these are not specific to aquaculture, but true for aquaculture. So I'll list them here. Production processing costs are high. They do compete when it comes to feed ingredients with other livestock industries. The regulatory compliance burden is relatively high, 3,000 to 5,000 per acre. There's competition from countries that have different production standards, and the standards that we're talking about come from the state government, the federal government, and sometimes local governments. Public perception issues based on misinformation, and so farmed fish sometimes gets a bad rap, and some of that's not based on scientific information, and so there's some marketing issues that the industry has to address. We have natural water resources that we could be using. However, our farmers have limited access to them. And then I think something that's true for all industries, if you look at COVID-19 and the fact that about 70% of seafood is sold to restaurants, sold through restaurants. And so when the restaurant market, was hit, the seafood industry was hit as well, and in particular our farmers. So some of the challenges that the industry faces that we can help with science and technology, and I think this is also a list that's true for many livestock and farm species. But developing new animal health management tools is incredibly important. In ARS, this is the largest part of our portfolio. Identifying new species for aquaculture. So what are the species out there that are wild caught that consumers enjoy, and how can we take on their figuring out their entire life cycle in the captive environment to make them an aquacultured species. Defining the nutrient requirements for each species that can be met with available, affordable, and sustainably sourced feed ingredients. This is incredibly important. If we're going to expand the industry, we have to be able to figure out how to feed all these fish. And so a very important question. I think Ken Overturf is going to tell us a little bit more about that. And then finally, at the bottom, I think these are issues that are important across the board: improved genetics, management practices, and production systems. And a lot of this is what we're talking about with the genome to phenome effort. And so I think just to make the point that aquaculture is also extremely relevant for this group. Always like to point to a publication from Dave Love and his colleagues at the Johns Hopkins School of Health, an analysis of $1 billion of aquaculture grants. And this went 15 years. 1990 to 2015. So Dave was a little frustrated. His grants weren't being funded, and so he wanted to take a look and see what was being funded. And so he looked across a whole lot of agencies and a lot of money that was spent in aquaculture research funding, and found that there was about a 37-fold return on investment for aquaculture federal research. And so we always appreciate that and want to keep that statistics right where it is. So in summary, before I turn it over to Ken and Jess, aquaculture is agriculture. A lot of the things that go into livestock and poultry farming is also true for aquaculture. We're talking about finfish, shellfish, and macroalgae, the seaweeds. There are many species that are in culture. Many are evolutionarily recently domesticated, and so a little bit different from traditional livestock. In some cases, we have industries that are still capturing wild broodstock and using them to get their juveniles. And so really still very much an industry in its infancy. There are many purposes, as I mentioned: foods and feeds, restoration efforts, conservation efforts, many types of environments. One thing in particular, I think that might be a little bit unique, there are many agencies that are involved, many federal agencies that are involved in aquaculture. USDA is very involved with many agencies, but also Commerce through NOAA, Fish and Wildlife Service, CPA, Army Corps, FDA, and many others. And also, when you look at states, sometimes aquaculture falls within a Department of Natural Resources, sometimes it falls within a Department of Agriculture. And so it can be kind of confusing. And so I think in terms of some of the challenges, figuring out a predictable regulatory framework for our nation would be something we could really benefit from. The biologies of these species really varies and can be very interesting. And in some ways, we're more comparable to other livestock species. And in some ways, we can use some of the approaches used in plant biology. So having high fecundity, chromosome set manipulation, and being able to make triploids and all different kinds of hybrids and things like that. So that's my summary. That's my introduction for aquaculture. Very much looking forward to these talks. So Dr. Jessica Moss-Small from the Virginia Institute of Marine Science is going to tell us a little bit about shellfish aquaculture and how it relates to genome to phenome. And then following that, we'll hear from Dr. Ken Overturf, who's with ARS in Hagerman, Idaho, and talk a little bit about genome to phenome as it relates to his work in rainbow trout. And so with that, I'd be happy to take any questions, and otherwise pass it on to Jessica and Ken.
Okay, thank you very much, Gerd. Very good overview and pointing out a lot of exciting opportunities for aquaculture. We do have a question from the chat, actually two questions from Dr. Allen. I'll take them one at a time. So what are the waste streams that are associated with aquaculture, and how do they compare with other agricultural systems?
What are the waste streams? So very similar, to livestock agriculture, right? So we have waste that's in the water. There are solids that can be captured, can be used as fertilizer. There are liquid waste that we need to treat. You want to make sure that you're returning the water to the environment just like you found it, just like it went into your system. And so with pond aquaculture, we're not talking about water that flows to other systems. And so you're using the pond ecology to manage your waste streams. For recirculating systems with the filters and things that are in place, there's an opportunity to capture those. For net pen aquaculture, where you are, feeding fish in the marine environment, you are putting in feeds. We need to make sure we're raising fish. One of the things about, finfish is that they have a really good feed conversion ratio. And so essentially a pound of feed is going to get you a pound of fish. So we try to minimize the waste streams that way. But also you have to be careful about your siting and making sure that as you add feeds and nutrients to the water, you're not going to do so in a way that it's going to have a negative impact on the ecosystem.
Actually, in that regard, second question from Dr. Allen is, in these natural systems, how does the aquaculture impact aquatic insects, both good, like dragonflies, and bad, like biting flies?
It's a great question. So that's not one that I'd thought about before. How do we, certainly in the open environment, there's going to be interaction with insects, maybe not so much in some of the closed, containment systems, but no, thank you for asking that. I'm not sure I have a great answer. Maybe Jessica and Ken will have a little bit more to add.
Okay, so maybe we can come back to that. Then a question from David Erdl. One of the reasons that's given for consuming more seafood is your omega-3s. How do farm-raised seafoods compare to wild sources of for omega-3 content? And then he goes on to say, I would expect lower if they're fed corn and soybean. And is there research looking at ways to maximize omega-3 content of farm-raised fish?
So that is a fantastic question. And so, yes, you see lower omega-3s in farm-raised fish. I mean, I guess I really should have started by saying they are what they eat. And if you can feed them a really high source of omega-3s, then they're going to have it in their flesh and they're going to match the wild. Typically that doesn't happen. And yes, we are looking at we do have research programs to look at how we can elevate the levels of omega-3s in farm products. And one of those projects is Ken. So that's spot on for this talk.
Okay, good. The question from Ryan Bartlemy. How skewed are the market values of aquaculture species due to morbidity and mortality in these aquatic species? For example, yellow perch are low on the list, but mortality rates are very high. So that impacts overall production.
Yeah, so certainly, having the on-farm losses due to disease or any kind of mortality event is important. We've seen, if you look at the news, we had one of the largest recirculating farms lose a whole quarter of production due to some unfortunate events recently. And so I'm not a marketing person, though. I think the question was, how does it skew the markets? And I can't really speak to that. I can tell you that fish are farmed by the numbers, and farmers have experience knowing on average what are their mortality events going to look like. They know there's going to be some. They know the amount of biomass they have in their system. They know how much they have to feed. And then we have ways of managing disease as well. And so there's always some risk involved in any venture, and including aquaculture for sure. But I think that's pretty much how they manage it, is fish by the numbers.
So would you say that disease is one of the major challenges in farming fish?
I think that's true. And, I've seen estimates, that farms at times can lose up to 30%. It's usually in the juvenile stage where they don't have as much investment. And so they kind of plan for that. And, if their numbers, in the good years where there aren't as many disease events, they may have to cull early on to, make sure they're not overstocking their systems, because overstocking is one way to surely lead to disease. So, farmers, have to be able to manage that, and, also later on use the tools they have to manage aquatic animal health as well.
Okay. Thank you very much. Well, time's moving on, so we'll have to move to the next speaker, but thanks, Gerd, and maybe you can chime in on some of the questions later on also.
Absolutely. Thank you.
Okay, so Jessica, if you want to share your presentation, and go ahead, please.
Sure, we'll see how this works.
Yeah, we're good.
Okay, and I'm gonna see if I can get the subtitles to work. So we'll see if that works. Okay, so it's almost noon for me here, but good morning for those of you who are out on the West Coast. And thanks for a great introduction from Gerd to aquaculture, and for mentioning oysters. So just going to give a brief overview of what I'm going to talk about. So why eat oysters, which is important. I'd like to focus on Chesapeake Bay, because that's where my main focus is. So I'll talk about the aquaculture industry here. In terms of phenome to genome, I'll talk about where we've come from mass selection for breeding to our family program, and where we are with genomic selection, which I'll give away the punchline. It's very early days still, but I'll update you as to where we are with that. I'd like to take a brief diversion and talk about Pacific oysters and some work that we're doing on the West Coast with Taylor shellfish, to talk about some other phenotypes that we're not working with exactly here on the East Coast. And then the last part is going to be about polyploids and where we are with polyploid breeding here at ABC. So why eat oysters? They're ecologically important. They maintain water quality, and they provide a really important habitat. Gerd gave a great introduction about how dietary guidelines suggest that we should double our seafood consumption. And so for oysters, it means that they're rich in iron, magnesium, phosphorus, copper, selenium, and vitamin B12. So it's a really great source of those. And as a protein source, oysters are shown to have less environmental impact than the beef or chicken industry. So for those who are up for it, I established here a brief poll. So if people swapped 10% of their meat consumption for oysters, the question here is, what would the greenhouse gas savings be if it was equivalent to taking how many cars off the road? So would that be, what do people think, 100,000, a million, or 10 million? I'll leave the poll open for a minute. We have a few brave souls. Looks like 1 million is the winner so far. Give it a couple more seconds. So why don't we end polling there? So it seems like most people thought it's around 1 million cars, but at least this in this link or this particular paper that I read suggests it's about 10 million cars per year, which is pretty exciting. So let's see if I can advance. There we go. So this is the species of interest, the eastern oyster or Chrysostrea virginica. Typically, farming was done using patent tongs by boat. This is the wild fishery that I'm referring to. These days, any farming or any harvesting that happens on reefs or on bottom is typically done by a dredge, which is the picture here in the bottom right-hand corner. So the history of oyster aquaculture really stems from the loss of wild harvest. It's an unfortunate but common mishandling of the fishery that occurred. So it was around the 1950s that the wild harvest was really in severe decline. And that was for a number of reasons, including habitat loss, overfishing, and disease. So we had two diseases that showed up here in the bay. Dermo, which it's the colloquial name for Perkinsus marinus, which is a protozoan parasite, as well as MSX also showed up here. Both of these pathogens prefer higher salinity waters. And in the late 1980s, we had a number of back-to-back drought years that really bumped the salinity up, and we had really severe losses, in the wild fishery. And unfortunately, in the late 1990s, this was, the loss of the wild fishery was very obvious, and a legislative initiative was put forth in the state of Virginia to, as I quote, do something about the oyster problem. And this is where the center here at the Virginia Institute of Marine Science came to be. So our acronym is ABC, and that stands for the Aquaculture, Genetics, and Breeding Technology Center. So, where is oysters and oyster aquaculture today? Oysters are on the rebound. The aquaculture industry is continuing to grow, even despite some, environmental hardships, and then now COVID. So just to put it in perspective, here on the East Coast, the 2018 farm gate value for Virginia shellfish aquaculture was about 60 million, and that includes hard clams and oysters. The numbers for oysters that year in particular were a bit low. We had 2 years of very low salinity here in the bay, and a lot of growers lost significant numbers of their crops, and so that was reflected in the number of market oysters sold. Virginia happens to be the first on the East Coast for oyster production, and most of that production comes from vertically integrated private hatcheries. There are two farming methods primarily used. One is on-bottom and one is off-bottom culture. So when we're talking on bottom, we're talking about spat-on shell or eyed larvae. So this is an eyed larvae. The eye is not an actual eye, but it's a light-sensing organ, that develops just as the animal is about to set. And what you're seeing here on the right-hand side of the screen, can you see my cursor? Okay, great. I'm not that familiar with Google Slides. This is a foot, so it's putting its foot out ready to set. And you can actually take larvae that you've spawned in a hatchery and collect them in the equivalent of a coffee filter, for example. And this is what people buy. So you buy eyed larvae by the million. This is what this bundle is here. These are all eyed larvae. And then folks have setting tanks. So these are cages full of shell. They then put these eyed larvae into the setting tank, allow the larvae to settle, grow for a few days. They'll then drain the tank, put it on a barge, and put it overboard onto leased ground. And that's where primarily you get a shucked product from. You'll get clumps of oysters that way. So the farmer would then go back a few years later and they would harvest those oysters, typically bringing them to a shucking house. Those would get shucked and the meats would be put in jars or frozen, etc. The other market is really for the half shell market, and this is the restaurant market. And that comes about by individuals purchasing typically small seed, even as small as 2 millimeters. They can then nursery them in upwellers such as this silo on the left or in the middle. This is called a FLEPSI or a floating upweller system until the seed becomes a bit larger. Then they'll put them overboard in either bottom cages like this, rack and bag. There's a lot of water column gear these days, as well as flip cages, long line systems, etc. So there's a lot of different ways that you can grow the single oysters. And again, what the goal is to have individual oysters for sale and shucking. So going from history, just a sort of a little bit more about what we are, and I'll put a link to our program in the chat in the end if people have interest in kind of seeing more about our research. So we have 2 hatchery sites. The one here on the left is where I am today in Gloucester Point, and then we have another one on the right-hand side, the Kaufman Aquaculture Center, which is about an hour away. We use about 5 to 6 different field locations for testing and our broodstock grow out. We've got about a dozen staff. Currently, we have a quantitative genetics collaboration with an individual and a group at CSIRO, based in Hobart, Australia. As I mentioned earlier, we actually also provide breeding advice and have a contract with Taylor Shellfish, which is the largest producer of shellfish in the United States, and they're based on the West Coast. So our main focus is on research. We provide a lot of advice to industry. We run a training program here as well, and we're a broodstock supplier. So by that, I mean, we don't sell seed and we don't sell eyed larvae out of here. What we do is we actually provide broodstock to hatcheries up and down the East Coast. And that's in the form of diploid broodstock or tetraploid broodstock, and I'll explain what the tetraploid is used for a little bit later. And that broodstock is actually intellectual property. So the hatcheries that get broodstock from us sign a licensing agreement, and then based on their production, they then return to our main university, which is the College of William & Mary, 15% of their seed cells, or $45 per million eyed larvae, which then comes back into the operating budget for our program to keep the research and the broodstock supply going. So it's sort of a positive feedback loop that way. And they pay for nothing upfront, I should add. And part of the service of us providing broodstock is we also provide a flow cytometry service. An individual asked before this started about triploids, and we have someone that performs flow cytometry here on staff that can verify the ploidy of seed as well as eyed larvae that's sold, whether it's diploid or triploid, and I'll speak about triploidy in a bit. So this is sort of our current menu of line offerings. This is found on our website as well, but we have a number of different diploid lines and tetraploid lines that are available to hatcheries. And they all were selected or have been developed by selection in different environments. If you think about Chesapeake Bay, which is our main sort of constituency, there's areas with very low salinity and low disease pressure. There's then high salinity areas that have high disease pressure and all the stresses that come with that. So we have a number of different lines that we've developed over the years, some from mass selection, which I'll talk about, and then from our family breeding program that have specific traits. So hatcheries will get a number of these from us. They'll then do the spawns and then sell them on to their growers. So where we are, kind of starting back from the beginning in mass selection. So I mentioned ABC was started in the late 1990s, but it was really in about 2004 that we started really thinking about genetics and how we would go about solving this problem with domestication and breeding. So at that point, we were building genetic stocks for disease resistance primarily. We brought in stocks from Delaware Bay that were resistant, generally speaking, to MSX, which is this bug here, Haplosporidium nelsoni, and then also brought stocks in from Louisiana, which were highly resistant to Perkinsus marinus or Dermo, and then started a very rigorous mass selection routine. I'll just step back for a minute. And from that, a number of lines were created. They have names like Debbie and Crossbreed and Lola and Hannah. We really like our acronyms here. From, during that mass selection program as well, and then propagating the lines, we also started our family program. And that, started by, as any good family program would, by bringing founding populations, so wild stocks from all over the bay, as well as selected populations, and then progeny testing them. So this is an example of our main progeny testing site. So we have 2 low salinity sites, one within Virginia, which is this site here, Louisetta, And then we also have a collaboration with the University of Maryland at Horn Point, and we have a long line system there in Cambridge. And that has a really great low salinity pressure. It gets as low as 5 to 6 parts per thousand for extended periods of time, which is really at the very low end of what oysters can tolerate. Our high salinity sites are here in the York River, and at times we've used this one, Lynn Haven, as well. So these are higher salinity. When I say high, not seaside salinities, so not 30 plus parts per thousand, but more moderate, upwards of 25 parts per thousand. And these have high disease pressure. Our gear types traditionally have been rack and bag, which is based on sort of the French system, where bags are strapped to these rebar racks in an intertidal area. But we also have in the past number of years been using Australian long lines, and this is actually from our site here in the York River. And those are excellent for testing, especially in terms of getting great heritability values for the traits of interest. So what are our phenotypes? Because this is the phenotype and genotype sort of talk. So not surprisingly, usual suspects, survival. We're reading for survival. Length, market size is typically about 75 millimeters. Total weight as well. And then also traits such as shape. So how round are the oysters? The market here likes something that's quite round, not something that's long and skinny. That cup depth, which we call height index. So you like a nice deep cup, but you also want it to be full of meat. And meat yield is very important, not only from the aesthetic standpoint of when you purchase an oyster at a restaurant, you want meat to fill that cup, but also from the shucking market standpoint, you want to get a lot of meat yield for every oyster that you open. So this is just a few pictures of us processing some of our families for phenotypes and data collection. So I know this is very busy, and I don't expect people to digest this, but just what we're getting in terms of heritabilities in some of these traits. So total weight, and these are across kind of low salinity sites and high salinity sites. Heritability for total weight, which correlates quite highly with length, about 0.36, so moderately heritable. Meat yield at times fluctuates in terms of heritability, but it's still something that we can breed for, definitely. Width index and height index generally also, so those shape characteristics have moderate heritabilities. And then survival. So underlying heritability for survival varies across sites and years, but generally it stays around about 0.25. So those are just examples of the genetic parameters associated with our traits of interest. And so we've been breeding with a desired gains approach. I will comment that we have two subpopulations in our diploid program, so a low salinity population and a high salinity population. And the reason for this is that survival does not correlate well across these two environments, and that's really because of disease. Other traits such as total weight and the shape characteristics and yield are highly correlated, but survival is not. So we have sort of two subpopulations and two separate indices that we use for breeding. And those traits and how we weight them is really based on their economic value. Probably a decade ago, we surveyed industry to see what was most important to them, and it really was a no-brainer. So it's things like survival and growth and yield, and so that's how we weight our traits accordingly. So this is sort of our yearly cycle now. It wasn't always like this. We used to hold animals in the field for many years. We used to collect data on them, 6 to 8 times during the course of their progeny test. But after, in 2012, we collected all the data that we had on our progeny test from 2004, looked at age correlations, site correlations, et cetera, and was really able to make our program more efficient. So we have our spawning, which is going on right now. Our progeny tests for diploids tend to last about 18 months, whereafter we collect data. We have an extensive breeding database that's become incredibly useful. It's vital to our program. With the help of our quantitative geneticists, we then get EBVs, establish what broodstock we're going to spawn the next year, develop the breeding matrix, and spawn again. But the question is then, how does this relate to an industry product? So though we aim to create about 65 high and low salinity families each year from the top performing families based on those weighted trait indices and the EBVs, We then generate terminal lines every year, and those get incrementally better year after year as the new progeny test data comes in. And if we weighted our traits accordingly, those genetic gains are continuing to increase. So we have a low salinity line, which we affectionately have called Lily, and we have a high salinity line called Henry. And so these are generated from our family program, whereas we also still propagate what we call our super lines, which were from mass selection. And the reason for that is those mass selected lines are really what industry had been used to and most comfortable with. We started offering Lily and Henry a few years ago, and they've taken some traction, I should say gained some traction, and also by doing some side-by-side deployments in the field and showing how equivalent or better these lines are, these family-based lines, relative to the mass selection lines, we've been able to encourage people to use them. So for the geneticists here in the presentation, you may be interested in kind of what our genetic gains have been. And this is in a recently published paper in aquaculture with my recently retired director, Stan Allen, and the quantitative geneticist I work with, Peter Kuby, in Australia. So on the left-hand side, A, these are the genetic trends for our low salinity subpopulation, and these are genetic trends on the right-hand side for our high salinity population. I should mention that we've had 3 phases of breeding with the family program. So from 2004 to 2010, we really were bringing in founders. So again, wild oysters from different geographic locations in the bay. From 2010 to 2014, we started doing more progeny testing, but hadn't really settled on the index or the weighting for the traits. So this is what we sort of call our R&D phase, and that's where things bounce around. I should say that this has all been standardized to the value for each trait in the wild population, sort of as a 0. So these are all relative to wild, unselected founders. So 2014 onwards is where we are now in our operational breeding phase. So green on both graphs is survival and the gains that we've made in survival. We've made a little bit better gains in our low salinity populations than we have on our high salinity, and that's likely because of the disease pressure. And I'll get to what we're doing a little bit more with disease later on in the talk. Total weight is the other one that we're pushing, and that's here. Meat yield has had an incremental increase, and then we're kind of just keeping an eye on the shape characteristics. We want to make sure that we don't make those negative, but things like height index and width index can also be highly a function of what the grower does in terms of husbandry. So we could do all that we could to put great genes for shape in these oysters, and if the grower has poor husbandry practices, they're going to drive them into the ground. So it's especially with floating gear these days and long line baskets, people can get beautiful cupped oysters. So that's why we really are just using these as sort of maintenance traits and more pushing more strongly those survival yields and growth traits. Some other traits that we're interested in, and these are in some cases collaborative efforts, we're looking at dermotolerance and resistance. So really, though we know that there's disease here, during our progeny tests, disease is very disease in terms of a field challenge is very heterogeneous, and it's hard. It's not something that's consistent year after year. So doing a lab challenge and correlating that to field survival is where we're at, and we're working with a USDA ARS geneticist at URI for that. We're also looking more at low salinity tolerance with lab challenges as well, with collaborators at University of Maryland. And we've also started using NIRS to look at the biochemistry differences, in families or in different times of year, looking at things like protein, lipids, glycogen. We've also been able to use NIRS technology to do a quantitative assay, which is very quick for dermo. So quickly, for those that don't know what NIRS is, it's near infrared spectroscopy. And in essence, you take a probe and pass it into a sample, which in this case would be your homogenized oyster sample. And depending on how those molecules vibrate, you get a fingerprint back or a spectra. Well, what you can then do, so if here's your NIRS spectra, and in this case, if this was a BCA assay for protein or this is a glycogen assay, you can calibrate your NIRS models based on actual benchtop values for those particular parameters of interest, and then be able to very quickly, just by using that NIRS probe, assess many samples in a short period of time for that specific sample or that specific parameter. So moving on to where we are with genomic selection. So we're about in year 2, and thanks COVID for slowing everything up, but we finally got some momentum. So this is a result of a large consortium project. So we have here on the East Coast what's called the Eastern Oyster Breeding Consortium. We're able to procure funding through the Atlantic States Marine Fisheries Commission and NOAA for a 5-year project called From Sequence to Consequence. And that, I should mention, it includes a number of individuals and a number of different research facilities and hatcheries from North Carolina, some individuals in the Gulf, all the way up to Maine. So it's a huge effort. A lot of herding cats, as you can imagine. I say that in the fondest way, in the fondest manner. So main objectives for this consortium project are to, not surprisingly, develop a highly efficient genotyping platform, then characterize selected lines, and also perform GWAS for important production traits. So things like dermoresistance, which I mentioned, resistance, or I should say tolerance to ocean acidification and hypoxia. In the Northeast, JOD, or juvenile oyster disease, is also an issue, so that is another trait of interest. And then, as I mentioned, low salinity tolerance. From that, once we have our genotyping tool in hand, estimate and apply genomic EBVs for genomic selection. So here at VIMS, also Rutgers is interested in doing that. And then evaluate genomic selection and identify high performance lines across growing regions. So, from where we are, and the map here has disappeared, apologies, in Google Slides, but from North Carolina, up to Delaware Bay. So where are we with this? So we first started with wanting to develop a 600K screening array, but before that we needed to get sequence data. So we have had a massive resequencing project from about 269 oysters, wild populations as well as selected lines along the East Coast. From that, found about 11 million SNPs. I should show you here, these are the populations, so wild samples from here in Maine, down here in North Carolina, as well as selected samples were screened on the 600K array. From that, we are now in the stage, and even yesterday, of finalizing our SNP selection for a 60K array. For VIMS data in particular, we mostly supplied tissue samples from our families, for our 4 or 5 different year classes of families for screening on the 600K array, and found that we could adequately be able to perform genomic selection with about 17,000 SNPs. The 17,000 SNPs that have been supplied for Thermo Fisher, who's designing the array, represent all haplotype blocks. We also had a few SNPs based on a small sample size that had, we did a small GWAS study. So those 150 or so SNPs are also going on the array for some association coverage. And this was through analysis done by some collaborators at CSIRO as well. So we're about roughly 8 weeks out from the actual physical arrays being shipped to our genomic services provider in San Diego. So unfortunately, I don't have a lot of data to show you yet, but within the next year, we'll be sequencing about probably 6,000 to 10,000 samples, and be well on our way for seeing how genomic selection works in our program here, what sort of values we get in terms of accuracies, how much more efficient our gains can be, etc. So I'm going to step away from Eastern oysters just for a minute as I look longingly at these pictures from the West Coast. I haven't been out in a few years because of COVID, but we've had a relationship with Taylor Shellfish out on the West Coast. So they produce a ton of different shellfish species between mussels and clams, gooey duck. They do some Eastern oyster production, but their main production is Pacific oysters, Chrysostrea gigas. I wanted to show a picture of just one of their freezers here on the bottom right. When I was there a few years ago, I was told that 3 tractor trailer loads of product go out of SeaTac Airport 365 days a year. mostly destined for Asia. And if you believe it, most of these beautiful oysters go for oyster sauce, which is sad to think, but there's a great market there. So they've been working with Oregon State University for quite a long time with the molluscan broodstock program there, and utilizing pods that have come from the MVP program. But the owners of this company were really interested in having more control over their traits, where their traits were going, etc. And so we started a dialogue with them a few years ago about providing them breeding advice and actually have a formal contract with them. So at Taylor, they're interested, as I should say, in the usual suspect traits. So they want oysters that grow and survive. They have an interesting hook in the hinge part of the shell that they also want to prevent because it makes it hard to open the oysters. But the thing that I want to talk about here is they want to make their family program POMS proof. And POMS is an affectionate word for oyster herpes virus. I think the Australians or the Kiwis decided to call it oyster herpes virus POMS instead of saying herpes because it's a little bit softer to say. So there is not the microvariant, which is the very virulent and lethal form of herpes virus. We do not have it here on the west coast of the US. However, they do have it in Australia and they have it in New Zealand. It also exists in Asia. Taylor also has a hatchery here in Hawaii, and so their concern is that it's the next thing that's going to hit the West Coast, and so they want to start breeding for herpes virus resistance now, so that they already have those genetics in their program. So what they've been doing is spawning families on the West Coast and then sending small seed for a virus challenge to a quarantine facility in New South Wales. So the seed are challenged in a bath challenge exposure, and then we get the data back for susceptibility, and it can include that in the EVV calculations. So just going to switch gears for a minute and do one more poll to keep people on their feet. Before I move on, I just want to ask, sort of silly, but I only eat oysters in R months because if people could respond to that, I know many people that I run into when I give tours or people come visit the hatchery say, well, I only eat oysters in R months. And I'm just curious as to why that might be. So people might respond to that before we move on. It'll give me a chance to have a drink too. So I like cold oysters too. I like cold and salty oysters. So I'm gonna give it a few more seconds. Okay, so it seems to be a tie here between I like cold oysters and I was told it was only safe to do so. So I'm going to end polling there. So I was told it was only safe to do so is very common, and I've heard that quite a lot. I would like to reiterate that especially farmed products are so closely regulated these days that it's really, stop share results. It's really safe to eat oysters at a restaurant any time of year. Also, the reason for the R months was that it was also because oysters were considered to have their best condition in the R months, so in colder waters. And that really has to do with spawning. How do I stop sharing the results? There you go. Which we've gotten around by developing the spawnless or the triploid oyster. So on the top, these are the same age and the meat quality. So these would be triploids compared to diploids here. So triploids, generally speaking, do not spawn out, which means they maintain their condition throughout the year. Also, because they don't dedicate those resources towards gametes per se, they also grow a lot faster. So the farmers can get them to market significantly faster than they can a diploid. There are some issues with triploids, which people feel free to ask about. But here in Chesapeake Bay, 90 to 95% of our industry is now based on triploid production. And so for those here on the field day that are concerned about polyploids, I'd just like to reiterate that polyploidy is rampant in agriculture. If you eat bananas, seedless cucumbers, seedless watermelon, you're eating polyploids. It's just not that common in animals. So we do have examples of it in fish and then in oysters. I won't get into how tetraploids are made. It's kind of a long process. Tetraploids are not natural. So tetraploids have 4 sets of chromosomes relative to our diploids, which is the native or the most natural form. The ability to make tetraploid oysters at one point was a patented protocol. It's not, the patent has run out, and you're free to do literature searches and figure out how to do it yourself. It takes some finesse, and it takes a number of years because you start with diploids, you then have to make chemical triploids, grow them up, spawn them, try to get chemical tetraploids before you get mated tetraploids. And once you have a mated tetraploid population, you can propagate those. So our hatcheries here in the US primarily or exclusively, as far as I'm aware, use tetraploids and diploids and mate them together to make triploids. If you're in New Zealand, for example, they do something called direct induction, where they do a chemical process on the spawning day where they go directly from diploids to triploids. But that's not often successful or very successful, and you can have batches that might be half diploid, half triploid. Whereas when you do the crosses with a tetraploid, and we always use sperm from the male and eggs from the female diploid to make a triploid, it's 100% successful for the most part. So that's how triploids are made. So because triploids are so important, we spend a long time breeding diploids, and we sort of have, we've been cranking the wheel on that, and we're confident where we're going. The next step, once we had a lot of tetraploids and different tetraploid populations in-house, is to start focusing on tetraploid breeding. So of course we would look at the usual suspects, but tetraploids are different. So things like sex ratio. As I mentioned, males are important for industry because they use the sperm. So if there was something that we could do about sex ratio in the broodstock that we provide, that would be great. Also, tetraploids like to revert. So the sperm, for example, will always be dihaploid in a tetraploid, but the somatic tissue could revert all the way to triploid in some cases. And so is there something that we can do about the amount of reversion or how intense that is? And then, not surprisingly, what's the correlation between, the traits in the tetraploids and the triploids? So we've been working on that for a couple of years. The things like weight have a high heritability, which is great, as well as survival. The one thing that we were also looking at was DNA content, and that there seems to be some issues with looking at DNA content, so we sort of abandoned that. The data really wasn't reliable, and estimates of additivity were problematic. However, looking at reversion intensity is something that we're still doing. Unfortunately, here for NINT is reversion intensity is negatively correlated to weight. So instead of having to run flow cytometry on a ton of tetraploids, we just have to pick the biggest ones, which is very convenient. So currently, in terms of tetraploids, our traits really that we're focusing on are survival and weight. And by looking at weight, we're also looking at reversion intensity. So that's all fine and well. But for quantitative needs, we really need to be able to link these two programs. So we've been breeding diploid families for years. We've been breeding tetraploid families for years. And so in the past few years, we've also been making triploid families and progeny testing those. Because the production organism is the triploid itself. So where we are now, actually next week, we'll be making diploid families, triploid families, and tetraploid families that all share gametes. So we'll be really able to get strong genetic correlations and be able to understand more what is the correlation between the traits in the triploid relative to the diploid parent and the tetraploid parent. That takes a little bit of funky math as well, but fortunately things like programs mentioned in this paper here allow us to calculate the G inverse. So, typical pedigree based on a diploid isn't hard. It is easy to calculate, and there's programs that can make the A inverse for you. But when you have parents of different ploidies or mixed ploidies, that can make things a little bit more difficult. So, fortunately, Hamilton et al. Did some work for us in advance, and is going to make the calculation of the correlations between tetraploids, diploids, and triploids a little bit easier. So that's where we are with that. And with that, I'll take any questions.
Hey, thank you very much, Jessica. Lots of very interesting work and lots of projects ongoing. We are running a little bit behind time, so but there are quite a number of questions in the chat. So maybe Jessica, I can ask you maybe to answer some of them.
Sure.
You have a chance in the chat while Ken is talking.
Sure. I'll go ahead
then-
and do that. No problem.
But then if there's time, we'll open up the questions later on. But turn it over to Ken. Ken, if you want to start sharing. Ken, you're muted. Okay. Okay, we see your slides.
Okay, so you can see the slides now.
Yeah.
And start. Okay. So you're seeing the full screen now?
Yes, we are.
Okay. All right. So mine's going to be a little different. It's going to encompass a lot of in the introduction, a lot of different species and an overview, and then we're going to drill down into specific physiological and biological mechanisms behind specific phenotypes that we found in our selection program. So, start this off. You heard from Caird, but as regarding the diversity of aquaculture species, not counting shellfish, crustaceans, algae, etc., according to the FAO, how many species of finfish are currently being commercially produced? And if Nicole, Phil, will go ahead and open up that poll. Which is, I can't see, is that running? Okay.
Yep.
So, right, so we'll let that go for a few seconds. All right. Now, people are varying. And looks like we're sticking there. Well, yeah, we're about there. All right. So we're 24 or 153. All right. And there. So we're a little bit more on 24. All right. Let's stop sharing results here. And so actually, according to FAO, there's 153 current finfish species. Now, unlike terrestrial agriculture, where there's like cattle, poultry, pigs, etc., you have a single species with multiple strains. We're dealing with multiple species with hardly any identified phenotypic separated strains to work with, okay? And in these species that we are working with, all right, again, some of them are mainly actively working on domestication and basically starting on growth, but they could be herbivorous, omnivorous, or carnivorous. They could be raised in saltwater, freshwater, or brackish. They can be in ponds or flow through. They could be raised in recirc, cold or warm water, and obviously the value of the animal that's being reared dictates the expenditure that is being used to improve it. Also, they could be live producers. They could produce a limited number of eggs, or we could have broadcast spawners that produce hundreds of thousands of eggs at a time. And then the species differ in ploidy, and this becomes quite complicated over evolutionary events. But it can be from diploid to up to 12 different copies. And then, as another nice little added complication, the sex of the fish can change over time according to the size of the animal or environmental conditions. But one thing that is interesting for a number of these species, though, is as opposed to terrestrial agriculture or livestock, a number of these have indeterminate growth, meaning that these animals will continue to grow throughout their entire lifespan and get bigger. All right. Even though typically for commercial practices, obviously there's a limited time frame that was being used in turning over production. But so why? According aquaculture is the fastest growing agricultural sector. Why is that? And part of it right now is, limited land available for terrestrial livestock agriculture, but also feed conversion ratios. aquaculture is very efficient in regards to pounds of feed necessary to create 1 pound of edible meat. And here's just basically a cartoon showing here, are comparing cattle, sheep, pigs, rabbits, and chickens to fish. And that's fish overall. Generally, salmonids now, we're about 1.1, 1.2 feed conversion ratios. So for about a pound going in, you get a pound coming out, which is quite good. Now, looking at these, you're like, well, what's different? And I think it's pretty obvious. Fish are raised in water as opposed to the rest of these. So why are finfish, so efficient, in growth? And again, being raised in water gives them 2 real main advantages. One, they're in a buoyant environment. They don't have to constantly expend energy to maintain a specific posture against gravity. There's a savings to that. Then also, fish are pacheliotherms, unlike homeotherms, so they're not having to expend energy also in maintaining a constant body temperature, which is good unless you're, obviously at extremes can be bad, but typically you're not going to try and raise an animal out of normal conditions. And then finally, vertebrates expend a substantial amount of energy in concentrating ammonia to uric acid to excrete as urea. Fish being in water can passively diffuse this ammonia across their gills into the environment. And depending on how you look at this, overall, these can be 30% or more energy savings as opposed to terrestrial. So where did I come into this, or our group? And this is, we're going to go back and we're determining some of the restraints or constraints on aquaculture. So as aquaculture has grown, the nutritionists were probably one of the initial groups working with that and trying to develop diets. And it wasn't that long ago when they were feeding raw horse meat to fish as feeds. Okay, but as it progressed, they found out fish meal and fish oil held all the optimum products or nutrients for creating a good diet. And in the early 1990s, as aquaculture was still growing, they only used about 12% of the annual fish meal and fish oil production. However, in the 2000s, we were now over 65% of fish meal and over 90% of the fish oil. And these are very finite quantities available. There's about 4,000 metric tons of fish meal available. And in years of like the El Nino, this could be even substantially less. So with the growth, these were non-sustainable sources. So even though they were primed, they were not going to be available for the expansion. So we went and began looking for sustainable protein and oil products. Typically we went to plants, and soybean meal being one of the most available has kind of led up to the top. But in looking at plants overall, there are some issues. And those include amino acid mineral balances, palatability, making that diet so they want to eat it, anti-nutrients, all right. And then going to the oils, which, I saw questions in the chat line, under the inability to efficiently convert plant lipids to the healthy omega-3s, EPA and DHA, which consumers like for their brain and heart healthy attributes. And then also when it reduced utilization of proteins or diets made from plant material leads to increased waste or biofouling, and this affects downstream systems. And we want to limit that obviously as much as possible. And then trying to improve this or evaluate it, there's very limited lines for research to start working with or to even determine physiologically what's going on by comparison studies. So early 2000s, it was dictated, our group to go ahead and start a selection program and see if we can improve rainbow trout for the performance or utilization of a plant-based diet. And plant-based diet now could be a huge term looking at it, as we're talking total 100% replacement, we're talking 50% replacement of fish meal, we're talking replacement with what's the formulation going to be? So there's a lot of things that go into that. But we started this initially with the integration of 9 different strains. These are both commercial and conservation strains. And by strains, basically they were taken and maintained in different geographical locations. Phenotypically, there's nothing identified, separating these, other than other coloration spots, etc. So we started that, and again, basically, and I'd like to say we were geniuses, but I think serendipitously there was a lot of luck involved in identifying, working with a nutritionist and getting a diet that we could use for selection purposes. And we started with this diet and looking at attributes that would be desirable for consumers, for the producers to go ahead and utilize these fish and the diets that were being produced with them. So we've been selecting for fish on an all plant diet. So there's no fish meal, there's no fish oil in these diets. And we started with 15% soybean meal in this, and we've increased the stringency to 20 to 25. We've increased the growth of these animals from 175 to about 400 grams, over this time period during our selection. But while we're doing this, nothing, especially when we can't zero down and specifically identify that trait and everything involved with it. So there's other phenotypic changes that come along, or biological changes that come along with the selection. And these involve changes in immunomodulation, amino acid transport, muscle accretion, and pathogen resistance. So this is what we're really going to, talk about a lot today in seeing how these correlate phenotypically, or how we can identify that and maybe utilize that through aquaculture in general and selection programs. So first off, looking at that, after about 3 or 4 rounds of selection, we compared our selected strain versus a commercial strain. And a commercial strain, or a non-selected strain that I'm going to talk about today, is basically a commercial strain used by producers that's been domesticated and probably selected for growth, but typically nothing else. And then conservation strains, or strains maintained by wildlife, fish and wildlife, etc., and they're maintained just for the highest level of diversity. And what we showed is that we were able to, through selection, increase the ability of our fish to grow on a plant-based diet versus commercial and conservation, and the protein retention efficiency was improved also. So that's a good start. All right. Looking a little bit further, we had cooperators, which were very helpful. And look, so stepping back a second here, feed probably consists of about 50% economically of your rearing costs, but your largest mortality costs are due to losses from disease. So working with our cooperators, they do disease testing for viruses, common and important fish viruses and bacteria. And what we found when we evaluated our families is that they showed a level of resistance that was greater than typically seen in commercial strains, and even in some selected strains. And initially, when we were looking at these, we saw a great amount of variation for the resistance, between the two and between the families. And we found that over time, this became a little bit more homogeneous, where we saw a lot less variation for the susceptibility to these pathogens in our families. So that was interesting, and obviously we want to find more out about that. Now looking at the performance for nutrient utilization, this is an experiment that was done within our group where we took rainbow trout and they were given protein blends by gavage, 5% of body weight. So basically, this is not a total formulated diet, but just the protein. And these were both fish meal and plant meal based proteins. And because a plant meal protein blend does not contain all the essential amino acids necessary for trout, we gave that with and without the amino acids. So what I'm showing here is that gavage fed 5% plant protein blend with amino acids. And then we took blood from the caudal vein at specific time points after feeding, and we evaluated the amino acids in the blood. This is typically what's been shown in published research before, commercial lines, where you see there's initial levels of these amino acids in the blood. It drops down, right? It drops down at about 6 hours, and then you see it rise up and peak again at about 18 hours. All right. Looking in our selected strain, we saw that initial uptick, but uptake, but then we saw a homogeneous decrease out to 18 hours, and not that uptick that we were seeing in commercial lines. And looking at this closer, I mean, in comparison, when you look here, so you'll see this uptick, and especially looking at lysine, you see a very large increase in that. Obviously, there's no feed coming in, so any of these amino acids being made available at this time point now is due to the animal having to go back out and break down tissue to get the amino acids available to complete certain specific metabolic acts that are necessary for the animal, which we're not seeing in our selected animals, in our selected fish. All right, looking at this a little bit closer, we evaluated a number of amino acid transporters. This is one that just immediate absorption of neutral amino acids. All right, and again, like I mentioned, we use that 5% plant-based protein blend with and without the amino acids. And interesting enough, looking at our non-selective commercial strain, we saw what we saw regarding the amino acids in the blood. These initial uptick. This is an expression here though. Uptick, then decline, and uptick again and decline. Selected looks the same. We saw the uptick and then a homogeneous decline with the presence of all amino acids. However, when all amino acids aren't present, basically we see no difference between the two strains. So basically everything has to be present for it to be able to efficiently utilize and process. And there's a strain difference according to that. So we know a number of these amino acid transporters are regulated by pH. All right, so we went back through and we looked at the intestines of these fish, both in the proximal, middle, and distal regions, and we evaluated the pH. Selected on fish meal, selected on plant meal, non-selected fish meal, non-selected plant meal. The only difference that we found according to this is the pH, increase in pH in the proximal intestine of the selected fish on the plant meal-based diet. So that's another point along the way. So from this and previous research, we knew that there was a lot going on in the intestine, and we wanted to evaluate that and look at it a little bit further. So working with a group out of Norway, actually Krogdal, who did a lot of soy-induced intestinal enteritis. It's shown here. This is intestinal inflammation from fish that are fed higher than normal levels of a soybean or just high levels of soybean meal in general. And a simple experiment where we did our 2x2, fed it 90 days, and then we sampled. All right, and this is just using a 15% soybean meal replacement. All right, and looking at the histological results of that, here is our non-selected or control animals on a fish meal, and we see the nice healthy microvilli, and everything looks good. Our selected fish on the fish meal, again, microvilli look good. Everything looks pretty healthy. However, the non-selected fish on the plant meal-based diet, we see the widening of the lamina propria, we see the clubbing of the villi drastically, and we see infiltration of immune cells, such that there's a lot of bad stuff going on here. However, in our selected fish on the plant meal-based diet, we don't see any of these effects at all. It looks basically the same as our non-selected fish on the fish meal. So that's favorable. And looking at that in more depth, we did the distal intestinal morphology, where we looked at a number of parameters in that. We wanted to look at cell turnover in that by PCNA staining. And then because we also know absorption and uptake of energy, besides amino acids, also looking at lipids, we looked at fatty acid binding protein in that. And not going through all of that, just quickly, here's our non-selected on plant meal versus selected on plant meal. What we showed in our initial histological slide, distal intestinal morphology was improved in our selected fish on the plant meal versus our non-selected. We saw in the non-selected fish on the plant meal, PCNA staining was significantly elevated, so there was a great amount of turnover here at the end of the ends going on here comparatively. And then we also saw reduced presence of fatty acid binding protein, which all of this is going to have an effect on nutrient uptake. So while we're doing this, nutritionists obviously they're continuing to look at different dietary ingredients, different formulations, etc. And they made a lot of progress. They had some really good formulations out there, and they tested these in labs across the world internationally, and they released a number of diets. And these diets went out to commercial producers, and there were issues long-term feeding these diets, where they saw that in certain number of species, they saw reduced growth, feed conversion ratios were more than doubling, and in higher levels of mortality. And which basically, in the short trials where these were evaluated, it's a much different when you're looking at this underproduction and production stress and over long periods of time. So now we have a model that we have a strain with a phenotype that we can use to go ahead and try to explore what's going on both nutritionally, genetically in fish. So, just basically taking our simple model 2x2, our non-selected fish on fish meal plant meal, or our selected on fish meal plant meal, we wanted to expand our analysis. And we did this quite a bit. So we maintained histology, but we added in, RNA-seq on number of tissues, proteomics, enzyme activity, metabolomics, and evaluation of the microbiome. Trying to get this, as I'll talk about what is going on together, holistically, not as single component, and how can either diet or genetics be used to manipulate that to make improvements for nutrient utilization, disease resistance, etc. So, but using this, it gives us a chance to look at the effects of selection, effects of dietary substitution, base genetic strain effects, and looking at chronic enteritis. And I want to thank Jason Abernathy for this slide here. All right. So, again, we've done this as opposed to that initial experiment. We do this, pretty much annually, and we want to look at it temporally. So we do evaluations at 3 months where we think it's pre-enteritis, looking at these animals, and at 7.5 months where we have enteritis starting to develop. And pretty typical, early on, we don't see much differences in, weight between these groups. However, at 7.5 months, typically see at least a 20% drop in the non-selected fish on a plant meal-based diet in comparison. 3 months, morphologically, the histology looks good in the non-selected fish on the plant meal. We do start to see some infiltration of immune cells starting even at that time. However, at 7.5 months, we give the full-blown enteritis and all the parameters that we discussed before. These parameters are shown down here. What we use as a typical evaluating total inflammatory cells, mucosal folds, lamina propria, and mucosa. Instead of breaking those out individually on a 1 to 5 scale, this is just the cumulative score of these for these fish. And as would be expected, our non-selected fish on the plant meal-based diet is significantly higher across the board on these. So this all fits into our model. All right, we have enteritis. We show it. We know it affects weight, nutrient absorption, utilization. It affects the morphology, and overall health of the intestine. So how much can we get out of this? And we started delving in, looking at, typically liver, muscle, etc. We did RNA-seq, and we've got a number of examples of evaluation of differential gene expression between strains and diets, effective enteritis and selection. Long non-coding sequences, we were actually able to detect a few sequences that reverse code for genes of interest. And then proteomics, we were able to using shotgun proteomics, generate a number of proteins we know that are differentially expressed according to diet and strain. And then recently we started looking at metabolomics and adding that into the group also. And where we know, the selected and non-selected individuals do have certain metabolites that are different. So instead of breaking these out individually, what does it mean overall? And we'll discuss that. Moving ahead, we also didn't forget about the the non-specific pathogen resistance, which I found really fascinating because we're not selecting for disease resistance. How can selecting for dietary utilization have an effect on pathogen resistance and where is it coming from. So this is a virus comparison here, and I've got a slide later where I'll show you how bacteria compared. But this is our selected, this is a selected commercial strain. This is a strain that's been selected for resistance to this pathogen. Here's our strain that's not selected, all right? And here's its relative resistance. And then here is the unselected commercial strain. And basically its resistance, which is minimal to this virus. And, looking over, this is comparing lysozyme levels, between these for the virus and for the bacteria. And, we see that there's significant differences for our selected strain versus that, not so much here in the virus. Also wanted to get a better idea immunologically what's going on, and we'd like to drill down onto this. There's a lot of things we'd like to do. Currently, we started with look at gene expression, so we had generated a panel of 17 genes, and on some of these initial time points where we looked at this over tissues, we looked at about 40,000 assays, evaluating stress, innate immune response, and adaptive immune response through expression. And this is again looking at 3 months and 7 months. And what we show, just in the expression of that, there's a significant change, and these overall in our non-selected strain, our commercial line on our plant mill diet versus our selected strain on either of these. And at 7 months, those differences are still there and even greater in some aspects. So we're still continuing to look at that. All right. Put it all together, and I know I'm not expecting you to really break this down, but just to give you an idea, we went ahead and did pathway analysis, which is, typical. It's difficult because we are looking at fish, and we tried using this where we did pathway analysis against the literature that's been published for fish. There's issues because there's not as much there, and then there's a lot of differences regarding the reportings of it, et cetera. So a lot of this comes back to evaluating against mammalian, basically human and mouse in general. But it is nice that the enteritis is a nice model actually for human ulcerative colitis and Crohn's disease. And so we see that, and we saw, a number of genes or protein or regulatory and threshold areas combining certain pathways. All right. So, starting to get a little better, helping to get a little better grasp, something else for us to focus on. But again, we know what's going on here is mainly in the intestine. So what else is going on in the intestine? The intestine is obviously it's important in digestion. It's also a very active immunomodulation organ, okay? And we know the microbiome plays a role in this. So now looking at the microbiome incorporated into this. On these initial studies, we evaluated the diet, okay? And obviously between the plant meal and the fish meal diets, we saw the biome, the bacteria we were able to detect of it was significantly different. We were able to show that even our inlet water was different than our tanks, but there was no differences between our tanks, all right? Now we're moving forward in this, looking at diet comparisons for the microbiome. The selected versus the non-selected fish on fish meal. There's no real difference in the microbiome between these groups. However, looking at the selected versus the non-selected on the plant meal-based diet, now we do see significant differences in the microbiome for these strains on these diets. And evaluating that by strain comparisons now, again, our non-selected fish, either on the plant-based or fish meal, again, we see significant differences in the microbiome according to the diet by that strain. However, in our selected fish, whether they're on the fish meal or the plant meal based diet, there is no significant differences between them. So physiologically, there's something that's been changed in our fish such that it supports the microbiome efficient, for the processing of dietary utilization, specific nutrient components. All right. Now, so we put all this, and we've been doing this and hoping all to get to this point, because what I've always wanted to do was try to get some of these data sets, large data sets and evaluations, and be able to compare them accordingly. And due to the biostatistical prowess of some recent postdocs, mainly Jason Abernathy and recently Jake Bledsoe, whose work I'm going to show here, we've been now able to look at, not even specific time points, but temporal transitional relationships between transcriptomes and the microbiome. So over time in the selected fish, how does the microbiome change, and how does that correspond to gene expression? And this is just showing time over time here, the enrichment of certain microorganisms in the selected fish compared to the non-selected fish over time. Now, when this is plotted with the transcriptome, we show there's correlated expression changes related to the microbiome seen in the animal, and it's specific to each of the strains. Okay. So, I mean, that's really what we're trying to drill down and start getting these big data sets now, and specifically, how does it all work together? What gene expression and microbiome, et cetera, or proteasome, what are the individual units that we can use to either evaluate diets or use in selection to improve for utilization? Now, we also went ahead and evaluated this looking at disease resistance, and just showing it here. All right, this is looking at a disease challenge between our selected fish and our non-selected fish for a specific bacterium. Our selected fish is about 30% more resistant, again, not being selected for resistance. And we show that accordingly, there was a transcriptional differences between the strains regarding when they're infected. And now we can we have comparatively evaluated the change in the microbiome over time during the infection between the two strains, and how the transcriptome and the microbiome changed in regards to the presence of a pathogen and its relative resistance, how it works to alleviate, susceptibility. So this is really the more recent right now, but this whole lot of implication in, again, genetically, what is important to select for that we can actually have resistance, not for a specific pathogen, but overall non-specific pathogen resistance, which would be much more valuable. And even also nutritionally, if we don't have that genetics on that, nutritionally, can we modify formulations such that we have nutrients that can help alleviate pathogen susceptibility? So pretty interested in that. So that's all I'm going to talk about right now on the plant protein selection base. But now we have this group. So now we've got lines of fish, stocks of fish, that can efficiently utilize an all plant protein diet. All right, and it was mentioned earlier on, well, what about EPA and DHA? Well, they can use other lipid sources to replace fish meal in these diets. All right, but when they couldn't test that before because they always had fish meal in the diets, and fish meal contains fish oil. So all right, so now we can do this without having fish oil. And we went ahead and we evaluated the ability for rainbow trout to biosynthesize and convert plant lipids, specifically alpha linolenic acid, to EPA and DHA and store that in the muscle. All right, and, we said there's variation for it and calculated the heritability. All right, so fish have that ability, or the rainbow trout do at least. All right, how can we improve it?
Okay, so Ken, sorry to interrupt, but we are running out of time, so I don't know how much more you have, but if you could try to wrap up.
Okay, I was wondering, yeah, basically just a couple more slides. We validated an assay. We were able to improve the trait 14% over a couple generations, enough that we were able to get individuals to accurately evaluate this. And there are other groups doing the selection also, using expression of certain enzymes for this. So we were evaluating it directly in the tissue. So we went ahead and we did proteomics and RNA-seq in muscle and liver, evaluating that. Didn't see really any changes in muscle related to that, to enzymatic into pathway regulation. Even in proteomics, we weren't really seeing the pathway. The only thing we did see, which was very interesting, was significant changes across groups regarding the binding protein. So basically the protein that feeds the pathway. So it's not actually being modified or regulated the pathway. It's being regulated at the level of feeding the pathway. And we were able to show that, incorporate that into pathway analysis, now allowing us to identify nodes or nexuses interactively for dietary utilization and disease resistance. And, that's our real goal here moving forward. And then finally, in summary, what we've shown and what a lot of people know, initially looking here, selecting for a specific phenotype can lead to multiple phenotypic changes, and it can, are you aware of what they are and can you utilize that? What we've really found here, stage and stringency of selection impacts the phenotype. And in this instance, tolerance to nutritional factors lead to physical manifestations regarding intestinal morphology, et cetera. I've already stated aquaculture is many diverse species, multiple untapped phenotypes that can be used to improve production. And we basically shown improved dietary utilization has the potential to reduce food costs, enhance pathogen resistance, and improve aquaculture effluence. And overall, obviously this wasn't done alone. I just at least want to acknowledge a number of individuals who assisted and were involved in this work. And I'll finish up there.
Okay, thank you very much. Very interesting work. And there's a number of questions, but I suggest because of time, and actually I have to teach in 5 minutes, so I have to drop out. But there is a Slack channel that Nicole, our program specialist, has set up. So you can continue the conversation there. And there may also be questions from others who are viewing the recordings. But yeah, I found it very interesting. And we've done work in pigs where we select for residual feed intake. So we are basically selecting for high growth under limited feeding. And I was wondering whether some of that is the reason for that, because we also find that the pigs that are more efficient, they are also more resilient. And it seems that they are able to are metabolically more flexible. They direct the resources where they're needed. And we can continue that conversation later on, but I was wondering whether that some of that is going on. I'd like to close because, like I said, I have to go teach. I'd like to close by thanking Caird and Jessica and Ken for a very interesting set of talks, giving us a lot of insight into what's going on, and interesting to see the connections with plant breeding. Is the question that came to my mind. Well, is there an opportunity for plant breeders to develop soybean lines that are better for fish meal? I don't know whether the market is big enough to justify that, but there's obviously some very good connections there. I want to thank everybody also for attending. Just the Slack channel, I think is ready if you want to continue the conversation. Remind you of future field days every third Wednesday of the month, 10:30 US Central. And there's another workshop that is being organized that starts on May 20, and that will focus on the intro to SNP data analysis. So look for that. Go to our website with a lot of other information. So I think I'll end it there, and thank everybody, and thank especially the speakers. And I'm going to get off and teach. Take care.